And I would like to welcome everyone and thank you for joining us. My name is Rebecca Lowe. I'm the Adult Program Coordinator here at the Lewis Public Library, and thank you for joining us this evening. And welcome to our series, and this is the last in our Spring 2025 series of Science and Society, Making Sense of the World Around Us. This lecture series is co-organized and moderated by Fred Dilla, Executive Director Emeritus of the American Institute of Physics and author of Scientific Journeys; Linda Dilla, former Public Information Officer at the Jefferson Laboratory and U.S. Department of Energy; and Colin Norman, the former news editor at Science. So with that, I am going to turn this over to Colin Norman. Thank you.
Thank you, Rebecca, and welcome everybody to, as Rebecca said, the last of our spring series of Science and Society talks. Today's talk is by Karen Lloyd. She's the Wrigley Chair of Environmental Studies and Professor of Earth Sciences at the University of Southern California. And as Rebecca mentioned, today is the day of publication of her new book, Intraterrestrials. Congratulations, by the way, Karen. It's a fascinating mixture of truly fascinating science, almost all of which has only been possible in the past 15 years or so with new technologies, and interspersed with hair-raising tales of how Karen and her colleagues collected samples in some of the least hospitable parts of the planet. It's a great read, and as you'll get a flavor of it from her talk today. As Rebecca mentioned, please put your questions in the chat. I will do my best to relay them to Karen at the end of her talk. So Karen, over to you.
Great, thank you very much. Make sure I do the screen share correctly. Share. Okay. Is it up?
It is. Looks great.
Okay. Thanks for the opportunity to speak to you all. I guess the introduction, Colin's introduction said who I am, but I'm going to tell you a little bit about the types of microorganisms that are buried deep in Earth's crust, or the ones that are using chemicals that come from the deep Earth, and how they're changing our understanding of how life can exist on Earth. I'm an active scientist, not primarily a writer. So I'm contributing to these discoveries together with my colleagues from around the world. And I'd like to bring you along with me on some of the adventures that we've been working on in some strange field locations. So let's start with a volcano. Not advancing. So this is a slide that won't advance. That's weird. Oh. Okay. So this is Poas volcano in Costa Rica. This is what it looked like on the day that I went into it in February of 2017. It's a nice day. There's no smoke coming up out of the volcano. You can see that it is a volcano because there's lots of dirt around here instead of any kind of vegetation. That's because it erupts every now and then, and none of the vegetation can really grow on it. So I had a field crew of about 20 people, and we really only chose the ones of us who absolutely needed to go down into this volcano because it does sometimes erupt without warning. So this is what we looked like going down into the volcano. It's kind of weird to think descending into a volcano rather than climbing up to one, but there's actually a visitor center at Poas. If some of you may have been there, it's a great place. You can't go there right now because it's actually erupting right now, so the visitor center is closed. But so here's all the folks at the visitor center watching us as we go down. This is me. We're wearing hard hats because all the small rocks could fall on you as you're descending into the volcano. So this is another picture of me in the volcano there. So that's us on our way down. The red spot here is actually like the throat of the volcano, not this lake. This lake is formed by deep waters that have filled with volcanic gases, and it is, and also rainwater that comes down, and it is basically battery acid. The pH of this thing is 0.85 on the day that I was there and measured it. So this is a giant, really placid, beautiful lake, but it will kill you if you fall in it. So it's a very scary place to be. When we got down into the crater, we could see the gases coming out from inside the volcano, and they form these little spicules and these gorgeous little sulfur crusts. So there's lots and lots of sulfur that comes in these gases and forms these beautiful precipitates, these minerals that you can form right in front of your eyes if you watch them form. This is pictures of me probably more terrified than I've ever been in my whole life. So this is one of my Italian colleagues, Donato Giovanelli, and this is also me. You can see that I'm surrounded by these volcanic gases. I'm wearing, my Costa Rican colleague was kind enough to lend me his glove, his heat glove, because the ground was 100 degrees and it was tilted at a very steep angle, and it was full of little scrabbly gravel. And if you fall, you can't stop yourself because the ground will burn your hands. But if you don't stop yourself, you'll go into this battery acid lake. So I was walking like a little mountain goat to collect samples. And if it sounds like I'm over dramatizing things, that could be, but about a month and a half after I went in this volcano, this happened. It erupted, and this is that little area right there is where I was standing at the edge of this volcano. And I will say that before this eruption happened, you can see it just keeps ejecting out of here, like continually. Before this eruption happened, there were signs that it was going to erupt. So, none of my colleagues were in this volcano when that eruption actually happened. So that was good. But a few of them were in pretty close to them. So you have to be really careful with these things. So why was I there? I'm a microbiologist. And what I want to know is how anything can live in a battery acid lake bathed in volcanic gases that explodes every now and then. And so one of the things we've learned about this strange life is basically how the essential features of what makes something alive have to shift just a little bit. Some assumptions that we make about how life persists have to be a little bit different when you're in pH, under pH 1. So quick primer on how all cells stay alive. So this is microbes in a volcano. This is my cells. This is plant cells. This is any cellular structure that's alive. We all move protons. So that's a hydrogen atom with an electron removed, which is just a proton, and that we pump these protons outside of our cells. And the energy that we use to do this, we get through respiration. So we get it through breathing oxygen. We move this proton outside the cell. This sets up a situation where we have more protons outside our cell than inside our cell. And this creates a gradient. And so any gradient that gets created is going to have pressure to go with the gradient and fall downhill. So the protons that are more abundant outside the cell want to rush back into the cell. And this sets up the conditions necessary to support our life, because we use this like water rushing down a waterfall. And it's almost like you can use a waterfall to turn like a mill to mill wheat, or to grind something, or to do some like mechanical function. We use this to make energy for the cell and everything that we need to do. So we use this property. It's called the proton motive force. So let's go back to the acid lake in the volcano. So acid means low pH. So pH 2, 3 is an acid, and this is pH 1. But pH, how we quantify it, it equals the minus log of the proton concentration. So what this means is that in an acid lake, you have a high concentrations of protons outside the cell and much higher than inside the cell. So you want this. You want more acid out here than less acid in here. So it's actually a good situation, right? Like we spend a lot of energy pushing protons out to create more acidity outside, but these guys live in an acid lake. So it's acidity is already free. So they should be able to get the protons sort of rushing in for free. But the problem with that is that we're not just talking about a slight gradient of protons. We're talking about so many protons outside the cell that it's like a massive waterfall. And so this is something that cells can't survive just if they let it ride. So even though they get this energy for free, it's like drinking out of a fire hose. They'll die. So they have to toughen their membranes to withstand this onslaught. And they can also create positive charges inside the cell that'll decrease this charge gradient. And then if that doesn't work, which that's usually not enough on its own, they just bail. They just are constantly tossing protons out of the cell. So I am not describing like a fun way to live, right? Like this is not something like, well, why do they live there? Yet we go all over the place and we find that there are microbes in volcanoes. So here's another volcano I went to. This is, even though it looks more dangerous, this actually was a much safer situation than in Poas. I was at a safe distance, although I did take this video with my iPhone. This is here a little bit of this is in Iceland. This is Fagradalsfjall volcano, and it is the most beautiful volcanic eruption I have ever seen in my life. You can kind of hear us laughing in the background of this video because it erupted about every 10 minutes. And every time it erupted, we just would stand there and like laugh with the beauty of the whole thing. This is what it looked like upon approach. So this is me, and you can see the volcano off in the distance. And then this right here is lava. So this is the lava that came from that volcano. It's this active front. This is not, you know, it looks kind of stable. This is hot and molten. This stuff is flowing. So you can see this is me right here, taking some of this new lava. I made I made this rock and brought it home to my kids. I was like, here's a rock that I made for you. And I'm not studying. So the lava itself does not contain microorganisms. As far as we know, this is too hot to support life. But I worked together with my geological colleagues to study the nature of the gases that come from this lava, because what we were there to look for is all of the microbes on the edges of it. So all these volcanic gases were seeding subsurface life in hot springs all around the volcano. And we wanted to know what was there. And so given that it's such a difficult place to live, why? Why would you live in a volcano? What is the point? What is the advantage for life? And the advantage is that they may present some challenges with the acidity and with exploding and being real hot, but they provide a lot of energy. And one of my deepest interests in the strange life inside Earth is that it ties biology to the fundamentals of energy in ways that we couldn't get at if we just studied the life that we see at the surface of the world. So what I mean is everything that we encounter in our daily lives, everything that we think of as biology, it's very diverse, plants, fungi, lots of things. We all use a single chemical equation. Oxygen reacts with carbon to make carbon dioxide. There's also photosynthesis, so I should say two, but in terms of respiration, photosynthesis is not a respiration, but we respire with oxygen to make carbon dioxide. Puppies do it, mosquitoes do it, and things that don't even look like animals but are, like sponges do it, and even things that are technically microbes but are a little bit bigger, so these are eukaryotic microbes called amoeba, these do it as well. The thing that's different about the intraterrestrials in the microbial world is that they deviate from what all of us at the surface can do. They can use, I say almost infinite number of equations because I don't know if it would be technically infinite, but it is probably uncountable. The pairings of chemicals that can be used to get energy by life inside Earth's crust greatly swamps anything we could ever know about from surface life. So you can look at a periodic table and pretty much everything in the middle is something that can be breathed by a microbe, and a lot of the things on the edges too. not the noble gases. We still don't know anything that can breathe a noble gas. But even within all these different elements, there's different compounds of those elements. So, it's not just that something can breathe copper and something else can breathe chromium, which is those are both chemical reactions that can give rise to life. It's that they can work with different oxidation states of those things. There's about, I think, 6 oxidation states of sulfur, and almost all of them can be breathed by some microbe somewhere. So this means that as a biologist, we don't often talk about fundamental laws, because biology is pretty specific, and ecology is hard to sort of make an equation to describe. But when we talk about the basics of energetics, we get to do the thing that we don't often get to do in biology, and distill everything down to one beautiful, simple little equation. And that is the Gibbs free energy equation. So this is the basic law of thermodynamics of thermodynamics at equilibrium. And what this is, I've written it for a specific reaction here, but what this is a measure of how much energy a cell gets, or life gets, to do whatever it wants to do: sing, walk, swim, whatever the thing is. And what this is made up of is, what is the nature of the chemicals that are reacting? Are they good at it? Do they give you a lot of energy or not? And then we also have to include this term for, are there a lot of those chemicals or a little amount of those chemicals? And these two factors are really the only things that go in to what can give rise to life. So, up at the surface, we only have one chemical reaction in terms of respiration: oxygen. That's pretty much it. So it's pretty dull. But in the subsurface, we get to see all these different array of things. So one of the places that I work that has a big array of different types of chemicals that can be used to support life are hydrothermal vents at the bottom of the ocean. So this is places in the ocean, usually where plates are pulling apart or shifting against each other, they look like volcanoes. They can be associated with volcanoes, but these are not volcanoes. These are superheated seawater that gets sucked down into Earth's crust, gets full of a lot of chemicals and a lot of metals that are very toxic to us, and shoot up out of these central chimneys. And I don't know if you can see these things around here, and if we were live and in the room, I would ask you what they are, but maybe you can think for yourselves just for a moment what you think these things are. Maybe maybe you've seen James Cameron's documentary and you know exactly what these things are, but they're worms. They're gigantic worms at the bottom of the ocean. They're 6 feet tall. They're taller than I am, and they do not have mouths or guts. They are totally dependent on microbes that live inside them. And so they are in a permanent symbiosis with chemicals coming from the deep earth, and they use the microbes inside them. They feed the chemicals to these microbes, and the microbes can then make food for the worm. And that's how the entire ecosystem keeps going. So when I talk about all these chemicals supporting subsurface life, there are surface organisms, these worms, that actually have harnessed the fact that these intraterrestrials, these inside the earth microbes, can do all these different chemical reactions. One of the ways that we go study hydrothermal vents is through submersibles. So this is a picture of the Alvin submersible. This is based out of Woods Hole, not too far from Delaware. And although I'm now realizing I should have put pictures up of my work out of Lewis, Delaware, I also work on a ship out of Delaware. Oh, sorry. Anyway, you live in an amazing place, those of you who are there. This is the submersible. This is the United States deepest diving submersible. It is currently rated to 6,500 meters water depth, which is a pretty impressive distance for a rather large submersible. It is hosted by the boat, the Atlantis. We load it with people. It carries 1 pilot and 2 scientists, and we load it and completely seal ourselves in while we're sitting on the deck to avoid any mishaps. And then this A-frame picks us up and sets us down in the water, and then lets us go, and we just free fall. We drop until we get to the seafloor and add some air ballast and slow down, and then do our work. This is what it looks like when we're preparing in the morning. This is about at dawn, the morning of a launch. We have this train track running on the deck of the ship, and the submersible is pulled back into the hangar. And here's where we can do work on it. All of the people, this person, that person, and that person are all pilots. So people take turns sort of piloting the submersible. If you're a pilot of the submersible, you also are responsible for its upkeep and engineering. So every pilot understands the interworking of every part of this submarine submersible, which gives me some confidence when I'm diving in it that they care so much for it. And then these are the deck crew. These are the people who are running the ship and sort of doing the same thing for the support ship. And both those teams work together to make launch and recovery happen safely. This is a close-up of what we call the basket. So this is like the front holding payload of the submersible. And Alvin has two arms to do work and to take video. And we set the scientists are responsible for the setup of this basket. So this is what we work on at night to reset it. We dive every day on a usually 2 or 3 week cruise. This is a picture of me setting up the basket for coring the next day. These are plexiglass cores with T-handles. These are good T-handles so the robotic arm can hold them and pick them up and take a sample. This is what it looks like when we load up. So this is myself and my PhD advisor back when I was a graduate student loading into Alvin. You walk across this little rickety stairs and you climb in and you close the inside. I don't know if you guys remember the Ocean Gate disaster, this really tragic disaster of a private submersible that was supposed to be diving to the Titanic and it imploded. And there were many engineering problems with that. It was a fundamentally different submarine than this one. This submersible is made of titanium and it's made in a sphere. And that one was carbon fiber, which was a totally untested and not approved way to build a submarine. But possibly the worst part about it was that you couldn't open it from the inside. So you had to be bolted in from the outside. So it's really nice to have control over getting out of the submarine. If you do pop up somewhere, you can get out on your own. So this is what it looks like inside. You can see this technology does not look like a Tesla. There's no LED in here. This is all the same rocker switches and analog connectors, and there's digital in there as well that was available in the 60s. Because if it ain't broke, don't fix it. This stuff works really, really well. And so they just keep it up and keep it going. And this is a picture of me with the porthole as we're starting to lose light. When you drop through the seafloor, pretty much anywhere, you of course lose light pretty quickly. Things get really dark. And then they light up again because all of the bioluminescent organisms that are filling the ocean sparkle when you hit them. So it's kind of like it's sparkly. It's not something that I would have thought of. I would not have thought of the deep sea as being sparkly, but it is. And it's really I don't study the stuff up in the water like that, but I think it's a magical place. This is what I do study. So this is these mats down here, these goopy looking mats. These are bacteria, and these are ones who are using the chemicals from these deep sea hydrothermal vents. This is another video. This is a video that I took. Sorry, that's the background sound of the submarine. You sort of listen to that all day when you're down there. But these are these worms, and you can see they sort of pull their feather heads in when we get close to them. These heads are there to collect the chemicals and bring them to the microbes that are inside them. But they're just teeming. They're all over the place. So, you know, we think of life as being so dependent on oxygen, but these organisms down here, well, the worms use the oxygen, but the microbes within them are doing other things entirely. So this is just a picture. We see lots and lots of animals that are supported by this. And this is what it looks like under launch. You can see we're not tethered to the ship. We drift quite far away. Swimmers have to dive around the submarine and make sure that it's safe and everything looks good before they give us a visual okay, and then they clear out. They go back to the ship with a small boat, and then we can release ballast and go down. Okay, so that's that's sort of the fun stuff. But what we do is we collect these samples and then we take them back in our laboratories. So that's one type of fun. Another type of fun is discovering things that nobody ever knew existed. And this is maybe sounds like too big of a thing to say, but it is absolutely true. What we discovered on that cruise, and we were not the only ones. Other people around the world were discovering this as well, that the tree of life, what is possible on Earth, is fundamentally different than what we knew before we started doing this work. So this is a tree of life. So this is the new classification for a tree of life that if you learned the 5 kingdoms, this has sort of gotten updated to the 3 domains. The archaea are here in the middle. The eukaryotes, this is us over here. This is what we knew before we started doing this work. Animals are here. This is us, we're animals. We are close to green plants, fungi, small protists, little amoeba, tiny little euglena, paramecium, tiny single-celled things are all here. But the archaea and the bacteria, which are microbes, are fundamentally different on the tree of life. They're totally separate groups for where we are. Almost all the pathogens that we fight, sore throats, pneumonia, these are all in the bacteria. For some reason, the archaea do not cause disease. We still don't know why. It's a massive mystery. I find it very intriguing. Someday maybe we'll figure out why. But they are fundamentally different, even though they look pretty similar under the microscope. So what we found after we came back up and we started looking in these samples, and we were using DNA to look and see what we found, is that the branches on the tree of life were actually more varied and deeper than what we knew before. So we thought we were going to be coming down, and sorry, I have an extra slide in here that I didn't mean to have. We thought we were going to be going down and maybe looking for some things that we already had. But what we found was that what we were finding was not things that anybody had ever discovered before. They were fundamentally different. We had no idea. That was in the early 2000s. And as you can imagine, when you discover totally new life forms on the planet, globally, all the scientists start working very hard to figure out what in the world they are, which we have. And so now one of these groups, this one, in fact, has actually now been brought into culture. People have it in a laboratory and they can study it, although it doesn't grow as easily as like E. coli and things like that. So it's still quite mysterious, but they've grown it well enough to take pictures of it. And 2 groups, as far as I know, have grown it. One in Tokyo in Japan and another group in Vienna in Austria. And they're beautiful. You learn in biology class that prokaryotes don't have complex structure. They're just kind of like bags of goo. And for the most part, they are. They can have flagella, but they don't have structure really. But these ones have structure. And this 500 nanometer scale bar here shows that they are ultra tiny. So this is the wavelength of visible light. And that's how big one of their cells are. And they have these structures that they can reach out and sort of grab each other with, which is just, I never would have thought that something so tiny could have such mechanical properties. But one reason why they can is that it turns out that these are actually our closest living relatives in the microbial world, as our progenitors in the microbial world. So they share a lot of the same elements, protein elements that give structure to their cells that we do for our cells, which we know have amazing structures, like axons and nerve cells have beautiful structures. And these little guys also share that because they're related to us, which is really fascinating. And we're just at the cusp of learning what these things can do. Okay, so now that we sort of got this like sniff of what's, what is life really like on Earth, how varied is it? It sort of makes you look at the world in sort of a different way. So do you guys recognize what planet this is? I never know if people get it right away, but I'll give you a second. I'll give you a hint on what planet this is. This is New Zealand. This is Earth. I think it is instructive to go to Google Maps, zoom all the way out, and flip the world around, because we almost never look at Earth from this angle. But I didn't do any tricks. Earth is a water planet. We cite that percentage, 74% of Earth's surface is covered in water, but you can actually get a view of Earth that looks like Neptune. It's so devoid of any land. So given this high prominence of the seafloor, we can add up the number of living microbial cells that are buried in the seafloor, and we get to a literally astronomical number. It's about 10 to the 29. To put this into context, this is about a third of the microbes on the planet are deeply buried inside our seafloor, which is about 10,000 times the number of stars in the whole universe. So it's really quite a big ecosystem. And what this means is that if we're thinking about, if we're thinking about questions of how life lives at the edge, how it uses different chemical reactions, how it uses energy in a different way than we thought it could, and how it's on deeper branches on the tree of life than we thought it could be, if we want to figure out what life is like, we have to study life inside Earth. So much of it may actually live inside. So the important thing that we've discovered about so much of this life that's just in sort of not the hydrothermal vents, not the worms, not the exciting places that have lots of energy, not the volcanoes. If for the quotidian life that's just settled on like everyday seafloor, just the boring parts of the ocean. I've heard it called that. I don't think there's any boring parts of the ocean, but the places that are just kind of mud, they don't have much energy at all. So if we get the equation back out, I made the delta G, this energy that's available, very tiny, because this means that the biosphere, this massive ecosystem, does not have enough energy to actually grow and divide. So when we see cells that are buried in these deep marine sediments, we find things that are potentially thousands of years old, or 100,000 years old, or possibly up to 1,000,000 years old. And so those strange microbes I was showing you with strange structures can probably live for up to 100,000 years or longer, which is, again, a fundamental shift in how life is. So this is all fun and, well, obviously there's value in just knowing that something is new, showing us new ways for life to be. But whenever we discover like new types of life, we want to ask the question, like, can they help us? So I don't know if you know, but Taq polymerase is one of the enzymes that allows us to look at DNA, and that was discovered in a hydrothermal vent. So these organisms have a lot to help us with, and one of the ways that they really help us is that, or hurt us, is that they are on the forefront of the ecological response to climate change. So I also do work in the Arctic. So I do this work in Svalbard, or some of this work in Svalbard, which is not technically in Norway. It's covered by the Treaty of Versailles that was signed after World War I, and it is administered by Norway. And it's extremely far north. It's at 79 degrees, which is basically the northern tip of Greenland. It's some of the northernmost land in the world. You can see I took these pictures from I took this picture from a plane flying over it. The glaciers sort of bulldoze all this sediments out onto the fjord. So there's just a lot of, it's a very active place. There's permafrost, there's lots of frozen soils, and everything is changing. The glaciers are retreating, the permafrost is thawing, and we are there to figure out how these microbes interact with all of that rapidly changing stuff. And also it is just a gorgeous place to be. This is the field team for one of our trips. This was not I was not on this trip, but this is my lab was representing me in this trip. So I showed this picture to emphasize just how international we are as a group. We have these three folks are from the United States, from Virginia and Tennessee, and another one from Tennessee. And we've got one person from the United Kingdom. Oh, sorry, she lives in the United Kingdom. She's Canadian, though. And then these three are from Russia. This work was done 2 days after the Russians started a war against Ukraine. So getting them out of Russia was very difficult because there were no flights. And I couldn't use my credit card to cover any of their travel. So they paid cash for a train to get out of Moscow to Helsinki, and I quickly got them a plane ticket out of Helsinki and into Norway. So they came and did field work with us and went back to Russia. So we were very lucky that they got out and could work with us. This one is from the United States. Tatiana lives in the United States, but she's originally from Ukraine and Russia. And these are two of my PhD students, and they're both from Nigeria. And my student Fumnaya said, I can't go to the Arctic. I'm African. And I was like, just put on more clothes. And she's like, oh, and now she loves it. And she's a full-on Arctic researcher. In fact, she just wrote a grant to go back and she was awarded that grant. So it's really, it's a place that entrances you and pulls you back. This is a picture of me in the Arctic, going up close to an iceberg. When we go out on the small boats, we have to wear survival suits in case we fall in. It's too cold. But it really is just a lovely, accessible place. The glaciers from the plane are also lovely, but the biggest danger that we have here are, I'll give you a second to guess what this is, whose paw print this is. This is a polar bear. There are more polar bears than people on our little island. And you can see this is a mama who came through with her cubs. And the day after she was gone, if there's a polar bear in the area, we stay away for sure. We don't court disaster. But I went up and took a picture. You could put maybe 2 or 3 of my hands in that paw print. So these are dangerous, dangerous animals, and they actually will hunt us. So when we're out and working, so this is me and my Russian colleague, Andre, and my English colleague, James, and this is me. And we're out there with this drill. So we're drilling permafrost through the snow, which is a arduous process. But at all times, we have to be armed just in case the disaster strikes. We are not out there to hurt the polar bears, but we have to be prepared in case they attack us. So we have to basically give up a scientist at all times to be scanning the horizon looking for a polar bear. And when you go up with full snow cover, it really is terrifying because every white thing on the white horizon seems like it's a polar bear. You can drive yourself crazy. But for the most part, you can kind of see them coming and you can move and you can avoid them and everything very peaceful. But that is something that we have to contend with in the Arctic. And that work is ongoing. We are seeing that these microbes are, as far as we can tell, pretty well poised to start degrading organic matter that is thawed. So what we're trying to figure out is whether this permafrost, the organic matter in this permafrost, can get returned to carbon dioxide. And at least as far as we can tell, it does seem to be returned pretty efficiently to carbon dioxide, but we are continuing to do that work. Another way that these deep organisms affect us is through, and I want to draw your attention to this because I think a lot of people have never heard of this, is deep sea mining. So mining is looking for metals inside Earth's crust. This is something that's really normal and absolutely necessary on land. There's no part of our lives that does not need metals such as iron, lanthanides, copper, manganese, iron, or I already said iron, all these things that are inside Earth. We need them for our batteries and for our magnets in our lives. But these metals also exist in the deep sea. And so there are current companies that are drilling, doing some tests to see if they can mine the seafloor to recover these things. I'm not going to say it's definitely bad, but I will say that I have not been very impressed with how it's happening. It's companies that sort of are operating in the dark. It's hard to go out and see what's happening down in the bottom of the ocean. I can personally attest to the fact that it is very hard to do work in the deep sea. Somebody could get away with a lot and you would never know it. But if this becomes a large-scale operation and happens in a way that the companies would actually make profit, then they're going to have to dredge a lot of the seafloor and collect these metals. And they'll pump this sediments up to the ship and separate out the metals from the sediment, and then they'll have to put the sediment back down into the water. And if they don't put it down far enough, it's going to clog up all the primary production here and stop making food for all the fish and stuff. So I'm mentioning it to educate as many people as possible that this is an industry that is currently in its nascence, and this may become a bigger issue as we move forward. And I think the more people who are educated about it, then the more social pressure we can apply to companies who are buying metals from deep sea mining operations to make sure that that's done in a sustainable way. And I'm not yet certain that it can be done in a sustainable way, but I'll keep my mind open. A maybe more positive industry that is, I think, quite hopeful, that also directly involves these weird little microbes that I'm studying inside Earth, is carbon capture and storage. So there are viable companies right now that are capturing carbon dioxide from point sources. So from industrial production of energy, to capture the CO2 that comes out of that, recondense it down, and push it back underground. And so this is actually a way to decrease emissions and potentially even draw down emissions, or draw down the partial pressure of carbon dioxide in our atmosphere, which is probably the biggest gas, definitely the biggest gas that's causing climate change right now. So the thing that is important to know about this process is that when you put carbon dioxide back down into the earth, that is a strategy that can work. But as we develop these technologies and as we move into these sectors, we need to be aware that we're not just putting it into an inert Tupperware container where you can just put things and then they'll stay. There's life down there already, and they may be starving and they may be ready to explode into action when you give them some carbon dioxide. So we need to be developing these technologies in concert with research in biogeochemistry to see, basically what the response is from these deep subsurface microbes. Now, I mentioned research and research funding. So I always acknowledge my funding sources. This is the whole enterprise of science is, I think, magical. I don't know. It's effective is what it is. Public funding for science has been incredibly effective at innovating and making the United States a leader, a worldwide leader in discovering new things about the world. So me personally, I've been funded by private foundations. The Simons Foundation and the Sloan Foundation have been wonderful, but the vast majority of my funding comes from public, federally funded agencies, like the National Science Foundation, which also funded this small offshoot group. The Department of Energy has funded a lot of my Arctic work, and NASA has funded this because NASA is interested in studying microbes that might possibly live on Mars or Europa. And so all of this public funding right now is being upended right now. We don't really know what's happening. The new proposals for the new funding for the National Science Foundation have been cut by 70%, which is going to destroy our ability to innovate in this country. I can't be too large in my sounding of the alarm bells of how much this is going to set us back in our ability to move forward as a nation. You cannot put innovation on the backs of companies, because companies have to make money. That's their job. And so they can be innovative. They can look to basic research and pluck from it the things that will make money. And that's wonderful. That system is great. But they can't pluck from it and make an iPhone and invent cool new things if we don't have the basic research that's fueling it. So this is something that I'm really hoping that Congress will see the light and recognize that we do need public funding for science. So I will end with this image. This is, if any of you are Disney buffs, or maybe have people in your lives who are Disney buffs. I have little kids and I've watched Frozen about 150 million times. This is not anything from Disney. This is my graduate student, Katie Seeps, in Svalbard. We went there in 2021, right in the middle of the pandemic. We had this long quarantine at a hotel in Oslo. It was just this arduous journey to get there. We finally get up to the Arctic, and she comes up to me, and in her backpack, she rolls out of her backpack. She's like, "Look, I brought an Elsa dress." was like, oh my God. So because it's the Arctic, we ran into a National Geographic photographer because that's where you run into people like that. And she was like, let me do a portrait of you at night. And so she was freezing her tail off and she tried to hold that pose, but she did end up in National Geographic and the European edition. So that was cool. But thank you so much for listening to me talk about these strange, weird microbes. This is really just the tip of the iceberg. There's a whole lot more to know about these guys. Anyway, happy to take questions.
Thank you, Karen. That was just a great talk. We've got a number of questions coming in. Let me just ask you one. Genetic sequencing is one of the keys to a lot of what you're doing. And as you point out very well in the book, a particular genetic sequencing technology, which came about 15, 20 years ago, absolutely revolutionized lots of fields, including ancient DNA and your field. And I wonder if you could just briefly talk about how sequencing has led to a lot of the things that you found out.
Yeah, in my everything you said is absolutely correct. In my mind, there are two revolutions. The first one is just the ability to look at DNA from the environment. That may sound like a small thing, but it opened our eyes up to the fact that these things exist. But we had our first step of being able to do that was very slow. It involved every DNA sequence that we got out of this thing was you take a sample from, a submarine or the permafrost and you crush it, take it back to lab, you crush it up, you chemically extract the DNA, and then you had to put it into a PCR amplifier and you put in primers and you amplify it up to a high quantity, and then you have to put it in E. coli and grow E. coli on a plate, pick an individual colony. It just took forever to get a single sequence. And so then the next big innovation in sequencing was from the companies and working off the backs of primary researchers in tandem. And that was the multiple parallel sequencing. And so now we go out and we collect a sample, we break it open, we collect the DNA, and we basically just put it directly into a sequencing machine. And the sequencing machine spits out just terabytes of DNA sequences. And from this, we can infer so much about the lives of these organisms.
And often you have the DNA sequence, but you don't know what the organisms are, right?
That's right. Yeah. And that it's a weird feeling. I say in the book, it's like discovering aliens, but the way you discover them is that you just get like a little slip from a machine that says, yes, this is an alien. It's like, give me something else, So a lot of the work that I and people like me do is try to put together the puzzle of what these aliens are actually doing.
Mm-hmm. A few of the questions that have come in have to do with whether the organisms that you're finding in the sediments, whether they can exist at the surface. And when you do bring those organisms to the surface, how do you keep them alive?
So mostly I want them dead for the most part. So I kill them usually when they come up because I'm going to crack them open and take their DNA out. So I'm not so concerned with keeping them alive. When we do want them alive, we just depend on the fact that a few of them will survive. There's also techniques for keeping them under pressure, and that can be helpful, but it's not the sort of silver bullet that you would imagine that it is. You can't just like apply pressure and then, you'll get lots of growth. You still have a lot of difficulty growing things. The first part of that, what was the first part of that question?
Whether they exist at the surface,
yeah.
whether the organisms exist at the surface.
Yeah. Do they exist at the surface? That's an interesting question that we're sort of not. It's hard to say that something doesn't exist somewhere because it could just be there in small quantities and we're missing it. But for the most part, a lot of these groups that we find in the subsurface, we don't find them at the surface. So it really is depending on where you are, because again, the subsurface has a lot of different ecosystems within it. And so the rules are different depending on where you are. But Um, there are subsurface specific deep branches on the tree of life, things that just are always down there.
Mm-hmm. And when you went down with Alvin to the deep sea hydrothermal vents, what are the conditions like there? Are they similar to the conditions in primordial Earth 3 or 4 billion years ago?
Yeah, quite a few people have posited that they are similar to how life began, and it's entirely possible. I mean, those ecosystems were on Earth around the same time that we think life may have started on Earth, although we're not quite sure when that happened. The earliest signs of life are at about 3.85 billion years, but Earth is 4.5 billion years old, and so we don't really know. Life could have started before then and just all the traces of it have been removed. But we definitely look to hydrothermal vents and other types of subsea floor environments as where you could be forming life on early Earth, because there's so many different gradients to exploit, and different chemicals to use. It just seems like a good incubator for life.
Hmm. And what implications do your findings about these organisms in these really uh, hostile environments have for searching for life elsewhere in the universe?
Um, for me, they give me great hope for finding life elsewhere in the universe. I think if we were relegated to just, if there was no life in the subsurface, I would just think, okay, well, life can only exist on the surface of planets. And when I look at pictures of Europa, I don't see any worms, I don't see any tractors. So there's nothing alive there. But we know that being on the surface is not the only thing that life can do. And so when I look at Europa, I imagine teeming life underneath its oceans. I mean, we've never gone through the ice on Europa. We have no idea what's under there. It's entirely possible there's whole ecosystems there. And the same on Mars. There could be life in the subsurface of Mars too.
Are there uh, are there plans to look at the subsurface of Europa?
So the Clipper mission, which is currently headed to Europa, got descoped. So that means that it did have a lander mission on it, and the lander mission got scrapped. What gets launched is a big mixture of political will, right place, right time, just a lot of things, and just the magic didn't work. So the Clipper mission is going to Europa, but nobody is actually going to land on Europa. But the plan was to land on Europa and to have a little drill that is got a hot nose. It's called the mole. And actually, I'm not sure if this was going to be on this mission or if this is a future mission, and it will basically melt its way down through the ice and sample the subsurface of Europa. Maybe
Uh,
someday.
a question about evolution of organisms that can live for hundreds of thousands of years. How does that work? Normally, when we think of things evolving, it's genetic mutations that are passed from generation to generation, and the ability of those to help you exist in particular environments. How does that work with organisms that are hundreds of thousands
Yeah.
of years old?
So it really challenges our conception of how that works. But we have to start with the premise that that is what's happening, because we'd have to have a really good reason to reject Darwinian evolution. There's no evidence that that's not happening. So if that is happening, then the way to evolve to do nothing for 100,000 years would be to whatever adaptation allows you to do that: toughened membranes, really great repair enzymes, and I think a lot of mechanisms, to intracellular mechanisms, to resist the urge to be fast, to just slow everything down and to like be in sort of a stasis state. So all of those things would have to be genetic adaptations. And what that means is that things that are surviving for 100,000 years or whatever have to have an advantage when they do start reproducing again. They have to be more abundant to pass their genes along to the next generation in the same way that natural selection always works. And so my idea, and this is a theory. It's really hard to prove, but it would work, is that when we have things that live for that long, we can start to bring in geological processes into their single lifespans. So an organism that lived that long, an ice age is only 30,000 years. So something that lived for 100,000 years would experience 3 ice ages in its life. And so after only 30,000 years, maybe the ice age sort of pushes it around and shoves it up, something about the ice moving in or the glaciers moving in like pops it out, and it can be around more energy, and then it can start growing. And so I think that there are geological processes that happen so infrequently that we wouldn't think of them as drivers for evolution. We can think of seasons as drivers for evolution, no problem. A tree can wait until summer to start making leaves because we know summer is going to come around every year. But if you can wait for 100,000 years, there's a lot of slower things that come around every now and then that could pass your genes along to the next generation.
Hmm. But by the way, did you find any organisms in the lake in Costa Rica? In
Yes.
the lake itself?
Lots and lots. Acidithiobacillus is the big winner. Sometimes it appears that is a monoculture of Acidithiobacillus. We can't quite figure out why that is, or if it just comes in after an eruption. The day that I was there, from the pictures that I showed, we actually found a very diverse group of organisms there. We found ferroplasma. We found lots of different organisms there.
Okay. Question is, how concerned are you about the various microbes and viruses in the defrosting permafrost?
That is a great question too. The short answer is I'm not very concerned, but I'll tell you why. So there was a really tragic event that sort of had an outsized effect on our understanding, but it was a very specific event. In Siberia, there was a community that had been using a local dump to put reindeer carcasses when a reindeer died. And so we know that carcasses of cows and reindeers, when they're buried in soil, can breed Bacillus anthracis, which is just a normal soil bacterium, but it's anthrax. It makes anthrax, which is highly deadly to humans in very low quantities. And so this is a known process. It wasn't a problem when everything was frozen, but with climate change and the reindeer dump starting to thaw, they actually had an outbreak of Bacillus anthracis, and I think 26 people died in that community. So it was that happened. There were pathogens Bacillus anthracis is not a pathogen, but it makes a toxin. There are things that can come alive and hurt us, but that was specific to reindeer carcasses. In terms of just natural permafrost, I can't think of anything in there that could get up the gumption to kill us. They're just kind of slow and dull. So I don't think they're going to kill us.
How deep have you found organisms, and is there a limit to the depth that you might be able to find them in sediments?
We can't say exactly how deep we've found living organisms just yet because it's kind of hard to know what depth you get samples from. The record is about 8 kilometers into the Earth's crust, which is 8 kilometers doesn't sound very far, if you're driving in a car or walking, but down straight into the earth is very difficult to get down that deep. But these were taken from rock fractures. So it's possible that the rock fractures were collecting water from either shallower or deeper than 8 kilometers, but somewhere in the vicinity of 8 kilometers. I do think that there's a limit on the depth inside Earth that you can find life as we know it, just because it's going to get real hot. So by the time you hit the mantle, it's going to be probably too hot for life everywhere. But the crust is, a couple hundred miles worth of, or a couple hundred kilometers worth of distance to go through. And we've never been through the entire Earth's crust. So there's already a lot of depth that we have left to explore.
Yeah, there was a infamous project 50 years or so ago called Project Mohole that was supposed to go through the crust. Unfortunately,
don't say it past tense. I know people who still want to go.
Yeah. In fact, I read somewhere that there are some plans being put together, but given today's funding environment, Not likely.
Well, in the States, it'll just be someone else. It won't be us.
Well, that's true. That's true. So,
Yeah,
while we're talking about funding, I hate to end on a down note. This has been such a fabulous talk, but how deeply are the current funding cuts and possible further ones, how are they affecting life in the research labs at universities?
I have felt the immediate effects of colleagues not wanting to come into this country because they're scared they're going to be turned away, or, people are very, a lot of the success of US science is that we have abundant resources that are very competitive. They are not easy to get, but when you get them, people want to come here. We can draw people. I'm sorry, I don't want to say anybody everywhere, anywhere, but we draw some of the greatest brains from all over the world, and they want to come here. And that has been absolutely integral to our success as scientists in this country. And I know that people are worried about coming here and don't want to come here because they feel that they'll be hurt if they come here. And that is a scary thing if it sticks around. If that becomes permanent, that's going to be worse than the funding. The funding cuts, yeah, we just won't get to discover things as much as we did before. So I'm just hoping that they won't happen. Those cuts have not happened yet. That's the hopeful thing. They're just proposed cuts. So maybe they won't happen.
Yeah, and Congress has traditionally been much more in favor of science than virtually any of the administrations for the past 30 or 40 years, at least when I was covering it. So there is
In
hope
in
that the Congress will.
in Republicans and Democrats, both parties have always historically been very supportive of basic science. So if this Republican government doesn't fund science, it will be quite a deviation from what Republicans have always been.
Yeah. Well, let's end on that hopeful note. Thank you so much. This has been really great. Just a wonderful talk. And traditionally, we ask people to unmute themselves and thank you for the presentation. So please go ahead and do that. And Karen, if you could just stay on the line for a little bit longer, there's a couple of things we want to talk about. Okay.
Great. Thank you so
Thank
much.
you.
Thank
Excellent.
you. Very
Karen.
interesting.
Thank you.
Thank you.
Bravo.
You.
Thank you. Thank you.
All right, everyone. Thank you so much for joining us tonight and also with the Spring 2025 Science and Society series. We will be back in the fall in September with our fall series. I shouldn't say we, it's Fred, Colin, and Linda are working very hard right now on putting that together. So keep an eye on our website to find out what it's going to be. So in the meantime, if you wouldn't mind logging off, you can go down to the bottom of your screen, lower right-hand corner, and just click on the leave button, and that would be great. We would really appreciate that. So although Colin, Karen, and Fred are going to stay on. Thank you. Thank you for joining us. So again,